Welcome to the course on Polymeric Biomaterials. We are in week 1 of lectures, and the topic of today's lecture is overview of polymeric biomaterials. So far what we have seen in the last couple of lectures is to look at biomaterials in general, look at the classes and generations of biomaterials, and we also saw how biomaterials of different types classes can be integrated into what we call devices or implants. And we saw examples, several examples deployed in the clinic of some of these devices as implants or prostheses. We also talked about why specific materials have been used with a little bit of history as well. As we move forward in today's lecture, our focus is going to be polymeric biomaterials. So in today's lecture, we will look at polymeric biomaterials around us first. What might surprise you is that a lot of the materials around you, as we will see in a very short while, in your room right now could possibly be used as polymeric biomaterials. Of course, there is a lot of science that has gone into it, but as we saw earlier, the starting point was industrial repurposing of industrial materials for biomedical applications. And then we will also look at some general features of polymers. We will look at more details of polymeric materials, the structure, function, property relationships, etc. later, but today we will look at some general features of polymers. We will also look at some properties of polymers that can help us classify them into different categories, and then we will look at the design application paradigm in polymers. This is a very important part of the course, and this will be a recurring theme throughout the course. And just to get a sense of why understanding structure property relationships is important for polymers, we will look at one case study of implant failure, where the incorrect use of a polymer led to some serious consequences when used as implants. And that will give us an idea for why it is important for us to study these in much detail and then use that knowledge to develop materials as part of devices that can function well and perform as expected. So with that, we will first begin with some general remarks on polymers. Polymers are not new by any means. They have been around for as long as life has existed in some sense. We have had polymers broadly that you might be familiar with along 3 categories. One is proteins, the category of proteins. Second is polysaccharides. The third is nucleic acids. Now these are what we would call naturally occurring polymers. Some examples of proteins that exist are collagen and silk, both of which we will look at in some detail in terms of their structure in the coming weeks. And in terms of polysaccharides, where the functionality is a sugar molecule or the monomer is a sugar molecule, there you have examples like cellulose and chitosan, where actually chitosan is a polycation with a charged polymer, and that has some interesting consequences. And of course, you must be aware of DNA and RNA, which are also long chain molecules but made of nucleic acids. Synthetic polymers, in contrast, a lot of the synthetic polymers that we know today are not more than 150 years old. Largely, as we saw in the evolution of biomaterials, derived from right around the pre and the post-war periods. So the wars led to a need and that led to a large spike in the production of new polymers, which we continue to use even today. So just to give you some historical context again, one of the first truly synthetic polymers that was ever made was this polymer called bakelite, which is a thermoset. We will look at what thermoset is in more detail again later, but for now what I am trying to do is introduce you to these words because when you learn any new course, it's important to get used to the jargon in the course. So, we will define these more formally later, but as you go through these lectures, try and get yourself familiar with some of these words, and as they repeat, you will get a much better sense of what they mean in specific contexts. So, bakelite is a phenol formaldehyde resin that was patented back in 1907, and like I said, it was the first truly synthetic polymer. It's what the thermoset means in this context is it's an irreversibly cross-linked polymeric structure. That is good enough to know for now, but why this is important was because this particular polymer had some very interesting heat resistance and electrical resistance properties, which means you could be using it in a lot of everyday applications, right from say things like kitchenware to electrical insulation and a variety of other household objects such as knobs, radio casings, toys and telephones. In fact, a lot of bakelite has also been used for costume jewelry, and even today it is used in wire insulation, brake pads and related automotive components. So, that was the starting point in some sense for polymers, and then we can now look at how another polymer, nylon, which was developed in the 1930s, continues to be used even today. Nylon has a very interesting history. Its production started right around the war. At that point, silk was big. Silk as a material for its exceptional mechanical properties was very big, but because of the war, the silk was not allowed to come from Japan to the US, and that led US to develop nylon in DuPont, and the scientist responsible for that was Wallace Carothers. In fact, the Carothers equation that we will look at later when we will study polymerization is named after him. So, as part of the effort at DuPont, nylon was developed and was used in applications such as being drawn into very strong fibers for parachute production during the World War. And even today, you have the bristles in your toothbrush are made of nylon. And of course, like I said, from a biomedical perspective, you have nylon sutures that are regularly used. Of course, nylon sutures are not resorbable, which means either they will stay there throughout or the surgeon will have to go in and remove the sutures at a later point. So, some other examples of polymeric biomaterials today, just as we have seen with nylon, which started in 1930s, and we continue to use them as biomaterials today. But if you notice, nylon's original application and intent of production was not for use in biomedical applications. So, now one can look at other examples of polymeric biomaterials, and all you have to do is look around you in the room that you are sitting in. You will likely see the following materials. One is polyethylene, which would be used in something like wire insulation, but as we saw in an earlier lecture, that these are also used as liners in hip implants. Of course, that is a slightly different version of a polyethylene, but it is ultra high molecular weight polyethylene. Now, cellulose, the primary polymer found in wood. So, you probably have wooden cabinets around you. But cellulose is also used in dialysis membranes. And likewise, if you take polyester, the first thing that comes to your mind probably is polyester shirt or polyester clothing. But you can also have polyesters like polyglycolic acid being used in orthopedic fixation screws. And lastly, polytetrafluoroethylene or PTFE. Most often you would hear this in the context of non-stick coating, but it is also used for the production of vascular access grafts. Of course, the form in which they are deployed compared to the household object is very different. They could be processed slightly differently, they may have different properties, but the essential chemistry has not changed much. So, the first example is that of the household object, and the second example is the biomedical or the clinical application. Now, that gives you a very good sense of how mankind has used materials around for specific biological applications. Of course, we have come a long way from there. In today's world, we can actually make a lot more interesting materials for clinical applications, but to be deployed in the clinic, it might still surprise you that many of the traditional materials still continue to be used, primarily because of safety considerations. Any newer material that you come up with, if the chemistry changes, if the properties change, they have to go through a cycle of evaluations. So, to avoid that, you can start with something that has already been approved and then see how to contextualize it for a specific application. And as you will see later in this lecture, if that is also not done correctly, you can fast track a specific biomaterial or a device for an application only to realize that it is not going to work in a different application at all. So, this all has to be done very carefully, and that is one reason why some of these materials still continue to be used despite the advent of newer and newer materials. So, you will find a lot of papers coming out on a regular basis, but in clinical practice you will see that some of the traditional materials still continue to rule the roost, and this is one of the reasons. It is because of safety, efficacy. These two things primarily drive clinical practice. Now, we will look at some general features of polymers. One of the things that you would have seen right away on the previous slide is we talked about polymers that could be used as liners for hip implants. We could be talking about polymers for use as vascular access grafts. We could be talking about polymers that could be used as orthopedic fixation screws. Now, just looking at the wide variety of applications, it should be very clear to you that polymers have a mind-boggling range of properties, because in one case you are talking about an articulating surface for the hip implant, where wear resistance becomes very important, and another case you are talking about pulsatile stresses, for example, that the material has to withstand. In another case you are talking about torsion and torque, for example in the case of screws, that the material has to withstand. Now, obviously one material cannot, it is very unlikely that one material can have all these features, but what is important to note is polymers represent a class of materials that come with a very wide range of properties that can be tuned. So, just as an example, you are looking at PTFE based vascular grafts here, and then bone cement for which polymethyl methacrylate is used. Two very different polymers, two very different materials, both of them are polymers. PTFE is polytetrafluoroethylene, and polymethyl methacrylate is plexiglass, as we saw in a previous lecture. These are very different materials with very different properties, but they are both polymers. And because they have very different properties, that also determines in what kind of applications they can be used. So, now you already see that there is a very specific relationship between the property of the material and the application. And that is a very important theme in this entire course, where you are looking at what kind of environment the material is going to be subjected to, what conditions it will have to withstand, what mechanical forces it will have to withstand, what pH it will have to be subject to. All these have to be accounted for from the application perspective to determine what properties are required for your device or biomaterial. Polymers also permit, like I said, large scale production and ease of processing, And one interesting thing about polymers is that they exhibit very important structure property relationships. So, just as I said that applications and the type of environment and the conditions under which the material will be deployed will dictate what kind of properties are demanded from the polymer. The properties themselves can be traced back to the structure of the polymer, and that structure itself can be talked about at multiple length scales. So, you can start off with a single molecule, which is the molecular structure, and then you can talk about a chain, you can talk about multiple chains interacting together, and so on. So, these are all things that we are going to look at in far more detail, but what I want to impress upon you in this lecture is not only that we can get a wide variety of properties, those can also be controlled and tuned based on how these polymers are processed, what their microstructure is, and so on. The other important feature for polymers is, for the same polymer, you may not be able to observe the same properties. One example that we saw is, we had to specify that the polyethylene used in the liner for the hip was ultra high molecular weight polyethylene, which is chemistry wise, if you think about it, it is not very different from the polyethylene bags that you probably use. But this ultra high molecular weight property or that adjective or that qualifier makes it very important when it has to be used as a liner. So, properties of these materials depend significantly on the source, processing conditions, and the structure, and all of those are important for eventually where these materials will be deployed in the clinical application. So, just as an example, this is a non-medical example or a non-biological example. These are common everyday materials that we see, baby diapers, and what you will see is that the main material in the diaper could be some combination of polyethylene and polypropylene, and then there is an elastic in the material which lets you tie it, and that is a type of a rubber. It is actually a segmented polyurethane, popularly known as spandex. That is the name. Interestingly, spandex is just an anagram of expands. So, if you do the anagram of expands, you will get spandex, and that should tell you something about the that is a mechanical property right there, where you can see that polyurethanes are specifically preferred for this application because they are extremely elastomeric, rubber like materials. They can expand quite a bit, making them to be used as elastic materials in the diaper. You also have an absorbing material which is sodium polyacrylate. Now, the sodium polyacrylate is a super absorbent gel material which is used in the diaper to absorb the secretions of the baby, and what is interesting about this is it is a cross-linked polymer which swells in water. Now, the cross-linking prevents immediate dissolution upon contact with water, and because it is cross-linked now, as it comes in contact with water, it is going to keep swelling and swelling and swelling, meaning it is going to absorb all the water that goes into it. We will look at hydrogel swelling much later in this course in much more detail from a completely thermodynamic perspective, and there we will ask questions like what causes this to swell, what is the thermodynamic factors, what are the thermodynamic factors that cause this to swell, and what prevents it from swelling forever, questions like that. And what are the contributions, enthalpic and entropic contributions that involve first the contact of water and then the swelling. So, these are questions we will look at in great detail, but it is important for you to understand that these kinds of materials that can absorb, super absorbent materials are largely cross-linked networks which can swell but not dissolve. So, having looked at some background about polymers in general and how they have been used and deployed in biomedical applications, we will now look at classification of polymers. And this classification, you can have multiple types based on what kind of polymer you are working with, what property you are interested in, and so on. So, I am just giving you a snapshot of the classification, and the idea here is that you are looking at polymers now along different categories based on what is of interest to you. For example, if you take source, you can have synthetic or naturally derived. If you take the synthesis route or the manner in which the polymer is synthesized, you can have step growth polymerization or chain growth polymerization. If you look at composition, for example, you can talk about things like polyester or polysaccharide. So, polyester we saw the example of polyglycolic acid, polysaccharide we saw the example of cellulose. Nylon, which we saw earlier, is an example of a polymer that is prepared using step growth synthesis. If you look at it from the perspective of architecture, you can talk about linear versus network polymers. If you look at it from the perspective of microstructure or crystallinity, you can talk about amorphous or semicrystalline polymers. If you think of mechanical properties, you can talk about thermoplastic or elastomer. And then if you think of degradation mechanisms, you can have hydrolytic, enzymatic, and so on. What I want to highlight here or emphasize here is this classification is by no means exhaustive, and the examples given are only a few under each. You can have more types of degradation. For example, you can classify polymers based on mechanical properties into other categories. You can have other types of architectures. Obviously, based on composition, you can have inorganic, for example, silicone based polymers, and so the list is endless. But idea here is to understand that how you classify polymers depends on what you are asking about the polymer. If it is the source that you are asking, where is the polymer derived from, then you classify it a certain way. If you are asking how is it synthesized, what is the mechanism of synthesis from a chemist perspective, then you can classify it a certain way. If you are more interested in the physical structure of the polymer, then you talk about architecture. If you are more interested in the microstructure and how the chains can, for example, pack and align, then you can have crystallinity. Or if you are interested more in bulk behavior, you can talk about mechanical properties. So, all of this goes back to what your interest is. And shown here are examples of two polymers where I have tried to highlight how the same polymer can be classified into different categories based on whether you are interested in source, composition, crystallinity, mechanical properties, etc. So, both of these are synthetic polymers, whereas PLGA is amorphous and hydrolytically degradable polyester prepared by ring opening copolymerization. PTFE is also synthetic, it is semicrystalline, it is a thermoplastic, it is a fluoropolymer prepared by free radical polymerization. So, if you look at the perspective of synthesis, then PLGA is prepared by ring opening copolymerization and PTFE is prepared by free radical polymerization. If you look at microstructure, PLGA is completely amorphous and PTFE is semicrystalline. if you look at what category do they belong to, PLG is a polyester, whereas PTFE is a fluoropolymer, and so on. And the other thing I want to also point out here is this list here is important for us because the rest of the next few weeks are going to depend upon how we go about systematically understanding each of these properties. Right from understanding polymer structure to polymer composition to polymer architecture to microstructure like crystallinity, and then looking at mechanical properties and degradation mechanisms. So, all of these things will be discussed in much greater detail. We will dwell upon each of these terms in a lot more detail as we go through the next few weeks. But like I said earlier, the idea is to get you exposed to some of these words right away at the beginning so that you get used to the jargon early on. And learning a course like this, it's important that you pick up the jargon without too much delay so that you can quickly try to understand the jargon in the context when it is used later. And before I proceed, I want to just highlight the design application paradigm in polymers. So, if you put polymeric biomaterials in the middle and you have structure property on one side and function performance on the other. Structure can determine the property, and structure property entity together determines how the polymer can function, and the function of the polymer determines how it performs in a biomedical application. In fact, structure property and function together determine how the material performs in a specific biomedical application. Now, if you look at it from, so that is from the biomaterial perspective. From the host perspective, if you remember the word biocompatibility, it has two components to it. Remember that it's not an intrinsic property of a polymer, de facto it's a property of a system. And you always have to talk about that in the context of a material interacting with a biological system. So, from a biomaterial perspective, if these things are important, from a host perspective, these things become important. You can talk about safety and toxicity, largely guided by structure and function, and host interactions guided by function and performance. So, safety and toxicity go hand in hand with structure property, host interactions go with function and performance. And of course, these two are also going to talk to each other, because if you have toxic effects, that is going to elicit an adverse immune response from the host, that in turn is going to have a negative effect on the biomaterial. So, they are all related in some sense, but this in one cartoon or one schematic is showing why it is important for us to understand structure property relationships in polymeric biomaterials and how that can be used to influence both the function and performance in a biomedical or a clinical application. So, just to drive home that point somewhat more dramatically, if you will, we ask this question about why are structure property function performance relationships important as part of discussing what I would call a case study, which involves looking at reconstruction of the TMJ using Proplast Teflon implant. Now, what is all this about? The TMJ is short for the temporomandibular joint, which is a small joint in front of ears that connects the jawbone to the skull. So, it is this joint that you are talking about here, and it is a very complex joint, very underappreciated in fact, with a lot of complex functions. Its correct functioning is going to let you do things like eat, laugh, chew, a host of other functions. TMJ diseases can occur, and they are characterized by pain in the jaw joint, which can lead also to limited mouth opening. You can have some clicking noises, popping, locking of the jaw. These are all things that can occur if somebody suffers from a TMJ condition. Now, at some point there was a proposal by a gentleman to use what he called a Proplast Teflon interpositional jaw implant to replace dysfunctional TMJ discs. So, the idea was to replace the TMJ disc with this new Proplast Teflon joint. Now, this is actually a combination of Teflon, which is nothing but the commercial name developed by DuPont for PTFE, which is polytetrafluoroethylene. So, Teflon FEP, FEP is fluorinated ethylene propylene resin. So, that is the resin that was made, and the idea was to see if you can coat that resin with a porous PTE, which this was called Proplast. It was patented at that point, and the idea was if you have this resin laminated with the porous PTFE or Proplast on top, the porosity would allow tissue ingrowth and thus could perhaps be enabling the material to bond better and function better. At least that was the thinking. And at least two different types of Proplast were prepared. One was Teflon and graphite, the other was Teflon and aluminum oxide. Everything looks great so far, everything looks like it is going to work, but something did not work, and that led to catastrophic failure. What happened? That is the Vitek implant case study. So, in 1967, Vitek Incorporation is established by a scientist, and in 1976, that scientist proposes the Vitek implant for TMJ reconstruction as described on the previous slide. What is interesting about this entire exercise is how the chain of events unfolded, and it is very instructive to look at the entire sequence of events because it tells a very, very important story which has very, very critical lessons for us in biomaterial design and biocompatibility. In the 70s and early 80s, the scientist who had started this company had put out a series of reports indicating positive effects of Proplast in unrelated applications. So, the two red flags right there. One is a lot of the reports came from the person who had established the company with very little independent validation by other scientists. More importantly, all of these positive effects that were claimed were not in the context of TMJ reconstruction, but it was in an unrelated application. In fact, at that point the TMJ was not even fully classified as a load bearing joint. And in 1983, the FDA provides 510K status to Proplast in the absence of animal studies. Now, just to put that in context, in 1976, the Medical Devices Amendment Act was passed based on devices that were developed before 1976. One could, in hindsight, seek permission from FDA based on what they called substantial equivalence. So, just to put that in context, this was the act that required the classification of materials or devices, to be more precise, into 3 classes based on the risk level. But what was interesting was Proplast was given permission to market a pre-cut disc as it was classified as a class 2 moderate risk device. Now, this was based on substantial equivalence that they claimed to have shown, the scientists claimed to have shown, based on another disc marketed prior to the 1976 Act. So, right away you see there is a lot of complication that is going on here, and at this juncture I would encourage you to pause and look up a little bit more about what 510K status means, how are the devices classified. So, I just gave you just a quick summary of what the classification is, but I would encourage you to look at it in more detail, and also what is considered substantial equivalence, because that is a very specific thing that we are looking at here. And as it turns out, the wording itself is not that specific, that the Proplast founder took advantage of it here. And in the 1980s, they get this approval to put it into patients, and this is even when they had not even tested it in animals. So back in the 80s, there were surgeons who were cutting these 1 centimeter square sheets in the operating room and suturing them into the TMJ joint. It was not until the late 80s that the issues started cropping up, which led to the FDA finally withdrawing the product itself. So the issues involved loss of temporal bone, presence of large soft tissue, and implant fragments. So this is probably the patient's worst nightmare, where they're implanting something into a patient and the patient is experiencing the implant coming off in pieces every now and then. To the point where, like I said, the FDA had to remove it from the market, and just to put the lid on the problem, actually literally they bury the implant in a landfill in Houston. So this is the story as it goes, or the timeline of events. What went wrong? So the devil as always lies in the details. So Vitek Incorporation informs FDA that it would market the implant based on the claim that it was substantially equivalent to an existing product, which was silicone sheeting, already in use as a TMJ implant. Now that was probably fine, but where things went wrong was how they showed substantial equivalence. For example, PTFE is based on a very different chemistry. The backbone is a hydrocarbon. Silicone is an inorganic polymer. So right away you see that you're not comparing apples to apples. Secondly, the way they were processed was also different. So not only is the chemistry different, the way it was processed was also different, which meant that any substantial equivalence that you can show is not going to work in reality, because both the structure and the processing are different. More importantly, all of the biocompatibility studies that were done, so-called biocompatibility studies, I have to put them in quotes, because as we saw, biocompatibility cannot be determined based on in vitro evaluation. They claim to have done in vivo evaluation using alveolar rich augmentation, which is a very different procedure. Although that also happens in the mouth, that's a very different procedure and a very different type of tissue, etc., compared to the TMJ. So if you actually wanted to show substantial equivalence, this was probably not the way to go about it. So it was combination of factors, right from the substantial equivalence not being related to the type of polymer, the processing, and not even the correct kind of application. So now you see that it becomes immensely crucial to define biocompatibility in the context of a specific application. Ability of a material, I am just recapitulating the definition of biocompatibility that we saw a couple of lectures ago. Ability of a material to perform with an appropriate host response in a specific application. So none of that was done here. And what was bizarre was no animal or biomechanical studies were done until after its introduction into clinical practice. And shown here is an example of that from the study, where you see this circle indicating enlarged mass fusing the mandible to the right base of the skull, where you see bone fusing, and you also see the yellow arrow marking impingement of the right maxillary tuberosity on the right mandible. So obviously problematic things that the patients would have had to suffer a lot of pain after these implants had gone in. And this happened because the PTFE's poor ability to the mechanical forces or the wear resistance in that region of the body. And just as a bolt from the past, if you recall Dr. John Charnley, who was looking at hip implants, and he was trying out PTFE initially, and they had initially not worked very well, and that's then how he moved to ultra high molecular weight polyethylene. In 1963, this is if you see here, 1967 or 1976 is when Vitek implant is proposed for TMJ. You know, a good 10 or more years before that, Dr. Charnley warns surgeons that tissue reactions are likely to follow the implantation of PTFE if this material is subjected to abrasion, and that these reactions may not manifest for 2 years. So, when you talk about biocompatibility, time also becomes important there. How long is the material intended to perform there, and will it do its intended job for that period of time? I think the lesson to be learnt here is that from this case study is that not only is it important to consider what kind of structure property you want for that application, but you also have to keep in mind the type of environment that the material is going to be placed in, the type of loading it's going to be subject to, the type of stresses it's going to be subject to, the pH, and a host of other factors before one can even claim, and of course the time period for which the material will be remaining in that position before one can claim that this material is biocompatible. So, that has to go all the way up to functionality and performance.